Okay, Assalamualaikum warahmatullahi wabarakatuh and good morning everyone. I am Nur Zalina binti Zakaria and today I would like to present our study which is Computational Insight into the interaction of phenolic compounds from the Chlorella sp microalgae against the S-shaped amyloid beta fibril. So basically, this is a computational study where we want to see the interaction between the phenolic compounds and also S-shaped amyloid beta fibril. Let's move to our research background. So this experiment was conducted for Alzheimer's disease. Alzheimer's disease, AD, is a common neurodegenerative disease characterized by the impaired cognitive abilities and memory loss. So AD is believed to be caused by amyloid plaques and also tau tangles. So the amyloid plaque is produced by the AB Fibril, where the AB fibrils are produced by the accumulation of amyloid beta peptide. So here is the AB peptide derived by the beta and gamma secretive cleavage from the AB precursor protein here. So the helical peptide here undergo a conformational transition from alpha helix to beta sheet structure and eventually forming AB fibril. So the AB fibril stick together and form the amyloid plaque. So amyloid plaque deposited between the neuron and it will disrupt the signaling. Okay, so now we move to the Chlorella sp microalgae. So Chlorella sp is a green microalgae which can be found in the fresh and salt water, and it has been applied in different various industry such as pharmaceutical, food and cosmetic due to their richness of high value of chemical compounds such as carotenoids, vitamins and also phenolic compound. So let's move to the phenolic compound first. So phenolic are chemicals consisting of hydroxyl attached to the aromatic hydrocarbon group. So here is the list of the phenolic compounds extracted and identified from the various Chlorella species. Okay, so Chlorella sp has shown to treat and manage TAD by targeting the oxidative stress, cholinesterase activity and B amyloid aggregation. Okay, so here we focus on the B amyloid. For the third point, it has been reported that the extracts from these two species inhibited the formation of AB fibril and also disaggregated the already formed AB fibril. However, they couldn't identify which compound involved in the inhibition and also disaggregation of AB fibril. So that's why we conducted this experiment, because we want to identify which compound involved in the inhibition and disaggregation activity. Okay, so here is the materials and method. We only use molecular docking method, which comprises into 3 steps. Okay, so the first one is the preparation of the initial structure. We downloaded our amyloid beta fibril from the PDB database and also phenolic compound from the PubChem database. Okay, the second step is the preparation of PDB-QT and parameter files. So we prepare the PDB-QT files by converting the PDB files and SDF files into PDB-QT. So this PDB-QT is a format accepted by Auto.Vina. And then we conducted our docking simulation using Auto.Vina. And finally, we chose the phenolic compound, the pose of phenolic compound based on the binding score. Okay, so now we move to result and discussion. The first one is the binding sites, where we can see from PyMol, which quite easier. So the binding site consists of 2 types. The first one is the pocket and the second one is the surface. And here we can see the first one, pocket 1, pocket 2 and pocket 3. And this one is the surface 1, surface 2 and surface 3. So pocket 1 consists of epigallocatechin here, and pocket 2 consists of chlorogenic acid, biocanin A, and also hydroxybenzoic acid. Pocket 3 consists of cinnamic acid here. Okay, and then the surface 1 consists of commeric acid, and then surface 2 consists of caffeic acid and gallic acid, and the surface 3, okay, here consists of ferulic acid. Okay, so that's basically summarize these two pages. Okay, so we move to the next slide. Okay, so the next slide, binding affinities and interactions. So here the compounds were listed from the strongest binding affinity to the lowest binding affinity. Okay, so as we can see, the binding affinity was stronger for the ligand that bound into the pocket compared to the binding affinity of the ligand that bound on the surface of the AB fibrils. And we also can see there are two types of interaction, which are hydrogen bonds and hydrophobic interactions. So here are the binding interaction maps. Okay, and then we move to the type of binding interaction. The first interaction is hydrogen bonds, which involve the carbonyl and hydroxyl of carboxyl group displayed by acid compounds. Carbonyl and hydroxyl of chromone, here chromone, displayed by biocanin A, and hydroxyl of chromine, here hydroxyl of chromine, displayed by epigallocatechin, and hydroxyl attached to the cyclic, here cyclic, displayed by chlorogenic acid, and phenyl ring, okay, hydroxyl attached to the phenyl ring here, displayed by epigallocatechin. Okay, the next one is the hydrophobic interaction, which consists of two types, alkyl and pi interactions. So alkyl interaction involve the alkyl groups of phenolic ring and also AB42 residues, whereas the pi interaction involve the electron from the pi system of aromatic ring, with sigma here forming pi sigma, or with alkyl forming pi alkyl, or with electron from the pi system also of aromatic ring, forming pi pi. Okay, both are pi pi, where pi pi stack and pi pi T shape. Okay, depending on their position. Here are the key residues that is important and involved in the intramolecular network for maintaining S-shaped AB42 fibril. The network consists of hydrogen bonds and hydrophobic interactions, composed of alkyl, pi sigma, and pi T shape. Besides the intramolecular network, the ASN and GLN ladder also important for maintaining the stacking of AB42 fibril. So as we can see, the asparagine here N27 and glutamine here Q15, the asparagine and glutamine side chain, can form hydrogen bonded ladders which contribute significantly to the stability of amyloid fibril. So based on our result, only 4 phenolic compounds bound on the surface of AB42 fibril displayed the interaction with the key residues mentioned before. Alright, so here the conclusion. The phenolic bound in the pocket displays stronger binding affinity than phenolic bound on the surface of AB42 fibril, and the binding interaction involve hydrogen bonding and also hydrophobic interactions. And 4 phenolic that lie on the surface of the AB42 fibril may destabilize and disaggregate the fibril structure due to their interaction with the intramolecular network and ASN, GLN ladders residues, which are really important residue in maintaining the S-shaped AB42 fibril. That's all from me. Thank you.